type of genetic material that Retroviridae have? What type of genetic material do retroviruses have?
RNA.
It's got to be RNA, because retro means go from RNA back to DNA.
Single strand.
Sense RNA.
They have positive ssRNA. RNA, good. And what kind of genetic material does herpes viridae have?
Double strand DNA.
Right. Okey-dokey. And I actually got some questions about the difference between genome and gene.
Oh, God.
Your genome is all your genes. The entire recipe book or cookbook. A gene is one recipe. So a gene is a segment of the DNA with a promoter and a terminator probably. That would be an operon. So a gene has a promoter and a terminator. That whole thing together is called an operon. And you have multiple genes in your genome. The gene is the genetic instruction for one trait. And that trait is usually a protein. So, you know, we do genetics, Punnett squares and all that stuff. We're like, oh, Johnny has blue eyes and his wife Mary has brown eyes. What color eyes are their kids going to have? Well, it works out pretty well in your Punnett squares, but actually your eye color is determined by not just one gene. It's determined by multiple genes. Not only multiple alleles, but multiple genes. Because look at all the different range of eye color. Some people have dark brown eyes, some people have lighter brown eyes. Some people have really blue eyes. They say Elizabeth Taylor actually had purple eyes. And other people have more greenish eyes. Some people have like a rich blue eye, and other people have a pale blue eye. So it's multiple proteins that code for your eye color, not really just one. But the browns definitely dominate over the blues. And it's always funny because I sometimes you'll see two brown-eyed parents with a blue-eyed child. That can happen. If you have two blue-eyed parents, you're not going to get a brown-eyed child. I don't know where that kid came from. But yeah.
I have a question on the last exam 3. This is back to the gene names.
Exam 3 hasn't closed yet, but go ahead.
But
just
yeah.
be careful because some people didn't take it yet.
So there's
Want to ask me later?
What is the genome? Okay, I'll see you later.
Okay.
Because
Yeah, but we'll have a question about gene and genome. And again, on your final you'll get it because that's general knowledge. Alrighty. Okay, let's talk about mutations. You get a lot of these kind of questions on exam 3 and you'll have them on your final also. So if I change in my genome one of the bases, one of the letters, what kind of mutation is that?
Silent.
A what? It's a point mutation. It's a base substitution. If you change one letter, it's a base substitution mutation. We also call it a point mutation because it happens at one point in the DNA. Now, if that point mutation, if that base substitution mutation results in a changed amino acid in the protein the gene codes for, then that's a missense mutation. If that change in the one letter, if that point mutation, if that base substitution mutation results in no amino acid, that's called a nonsense mutation because it makes no sense. Okie dokie. If I have a point mutation, I get the wrong letter in the wobble position or the third letter of a codon.
silent.
That's the silent mutation. If I get the same amino acid anyway, first of all, what do they call that? What do they refer to that as? What causes me to get the same amino acid even though I have a different third letter? Degeneracy of the genetic code. Good. And our genetic code is a triplet code because you have 3 letters making up a codon, and the codon is the genetic word for what?
Genetic code.
One codon is going to be translated into one amino acid. Good. What if I have a mutagen and that mutagen binds to the letter and masks it, covers it, hides it? So when the genome is being transcribed or copied for that matter, it skips a letter. What kind of mutation is that? Frame shift mutation. So, which is the worst kind of mutation? That one, frame shift mutation. And that does not occur naturally or spontaneously. Point mutations can occur spontaneously. The rate at which those mutations occur is relatively rare, but frame shift mutations do not occur spontaneously. Those would be induced by some mutagen, some agent's going to cause that to happen. Like the charcoal stripes on your steak, like the smoke in your cigarette. Not fully combusted organics causes that. Those are called benzpyrenes. Also, aflatoxins. Aflatoxins can come from aspergillus mold. And aspergillus mold sometimes will grow in stored grain. You know what a silo is? When they store their grain in there. And if mold gets in there of a certain kind, aspergillus flavus, I think it is, it can produce aflatoxins, and then the aflatoxins could be, say, in the peanuts. And then if they make peanut butter out of it, naturally, just grind up the peanuts in the health food store, there could be aflatoxins in it. This is one of the reasons why they irradiate food. Because if you irradiate it, it'll kill the toxins. So they can irradiate the peanuts before they make peanut butter out of it. Right? So when you irradiate food, it's to clean it. Kills viruses, toxins, bacteria, mold, whatever could be contaminating the food. Alrighty, let's see. Are all mutations bad?
No.
No. Some mutations are actually good. If it helps the organism survive, it's a good thing. And, okay. Okay. Let's talk about the difference between selective agar and differential agar. I think people still haven't wrapped their mind around this. Special media or special agars has special ingredients added to it. If the special ingredients make it selective, that means you've added ingredients that select for the growth of one type of bacteria over any other. So you add a special ingredient that helps a certain kind of bacteria. That special ingredient might also inhibit some others, but it doesn't have to. All it has to do is help one grow better than the others. And then that one is going to take over by competition. You've seen that happen on your own Petri plates, where you have maybe E. coli mixed with Staphylococcus epidermidis, and when it grows, only the E. coli grows, because it wiped out the Staph epidermidis. So when you grow bacteria side by side on a Petri plate, unless you can separate them away from each other and they have their own little space, there's competition there. And so one type might grow over another type. But when you add an ingredient that helps one type, then it's going to be even stronger in competition, so it might totally wipe out the other type. So selective agar helps promote the growth of one type over any other type. There might also be an ingredient that inhibits the other type, but it doesn't always have to be. As long as it helps one, that's enough. And then your differential agar is usually selective to begin with, but then the differential ingredient is something that one bacterium can use that maybe others can't use. And that ingredient is linked to a dye or something that causes a color change. So differential agars are usually colored, and you're looking for a color change, because the color change means that a certain type of bacteria has used a special ingredient. And when you get that certain color, then you're like, oh, I know what that is, or I know it can do this, so I can narrow down my identification. What did we use the API 20E test strip for? Okay, we use it for Enterobacteriaceae, but what were we doing with Enterobacteriaceae? Right, we're trying to differentiate bacteria. So your API test strip is all little differential, not augers, they're just little differential, what would you call it, liquid medium. And then you added your bacteria to it, right? So you add the bacteria and the liquid, and the ingredient was already in the little cupule. So those are all little differential tests you're doing in your API test strip. So when you do these differential tests, what are you looking for? What did the bacteria do in those little cups?
Change color.
They had some kind of a metabolic reaction, or maybe they didn't have a certain kind of metabolic reaction. So we're identifying bacterium based on their metabolic reactions that they can do. And what helps them run a certain metabolic reaction? Enzymes. So I'm looking for, if this bacterium, I get a positive, and a negative, and a negative, that means it's got a gene for this and a gene for that. It's got the enzyme to run this reaction, enzyme to run that reaction, but it doesn't have a gene for this enzyme to run that reaction. It doesn't have a, you know what I mean? And different bacterium is going to give you a different pattern because he's got different enzymes for running different reactions. Because each enzyme is specific to the reaction that it runs. Alrighty, I think you remember how to make a slide, right? You put your little drop of water on your slide, you mix your bacteria into it, you let it dry, and then what do? Heat fix it. Why?
To stick here.
Make it stick to the slide, so hopefully when you dye it and rinse it, doesn't rinse it down the drain. With the broth cultures, it's a little tricky. Sometimes that broth is so slippery and that when it dries in the broth takes up the water and slides right off. So making slides from the broth, I will admit, is very inconsistent. Sometimes it works, sometimes it doesn't. Alrighty.